Howdy. Uh, I'm pleased to announce today's seminar speaker, Dr. Daniel Koenig. He completed his undergraduate studies in biochemistry and cell biology at UC San Diego and PhD in genetics from UC Davis, where he worked on understanding the molecular mechanism responsible for interspecific shift in leaf shape. After his PhD, he was awarded a Human Frontier Science Program postdoctoral fellowship to work with Detlef Wiegel at Max Planck Institute for Biology in Tübingen, Germany, where he worked on understanding how the evolution of plant immunity was a major driver of genome evolution in Brassicaceae family. After this, he moved to UC Riverside, where he started his own research group to study the genetic basis of crop adaptation. So please join me in extending a warm welcome to Dr. Koenig. Thank you.
Okay, let me click this on real quick. So I didn't know that that was going to be read out loud. But luckily I didn't say anything too self-congratulatory. All right, so thank you very much for, can everybody hear me? All right, so thank you very much for the invitation. It's really fun to be able to come here and meet both graduate students and faculty. Especially for me, it's fun because this department has both basic and applied focuses, and it's really fun to think about the challenge of applying some of the basic work that my background really has led me to study in the past in the real world where things get a lot more complicated. And so I really appreciate this opportunity to share some of our work and really the hard work of the folks who work in my lab. So thank you for that. Today I'm going to tell you a little bit about the work that we are doing to try to understand the process of plant adaptation. Let's see if this works. All right. All right, so before I get started, I want to acknowledge the folks who really do the hard work that makes this possible. So the folks in the lab currently are shown on the left, but this project really started some years ago and was led by a postdoc in the lab at the time, Jacob Landis, as well as Angelica Garcio, who was a technician in the lab. So they really did the bulk of the initial work on this project. Many other people helped contribute along the way. And then at the end, I'll tell you some newer work that's only partially completed from a graduate student in the lab, Jill Marzolino. This work wouldn't have been possible without some really important collaborators. So here, Peter Morell at the University of Minnesota, who's an expert on barley genetics and evolution, as well as Cal Qualset, who is a retired breeder at UC Davis, and his role I'll talk about in a minute. And then we have funding from various sources. For the work I'm going to talk about today, most of the funding comes from NSF, from a plant genome research career award that I received some years ago. All right, now that I've acknowledged the folks in the lab, I'll try to introduce this problem that we are interested in. So we are really interested in general in how plants adapt to various environments, and especially how rapid adaptation occurs. So when the environment changes really quickly, how do plants deal with this? And one of the most spectacular examples of rapid adaptation is really the spread of crops after their domestication. So crops are typically domesticated in a relatively limited range. Here I'm showing Central Asia, which includes the Fertile Crescent, where a number of really important crops were domesticated some time ago. And today I'm going to talk about barley, which is thought to have been domesticated a couple times in the Fertile Crescent some 10 to 12,000 years ago. And it really kind of hung out in Central Asia for a while after that. It didn't really spread that much until about 3,000 years ago, when its distribution really exploded. It moved to become a major important source of human and livestock nutrition throughout Eurasia and North Africa. And of course, as many of us think about it today, barley is really important for the production of alcoholic beverages. But it remains today the fourth most important grain crop globally, and is still used for animal feed, and in some places for human nutrition, although less so. All right, so when barley moved from this relatively limited geographic range into all these new environments, it had to rapidly evolve to be able to survive in these new environments. So I probably don't have to tell you that northern Sweden is not too similar to the Nile region of Egypt with respect to climate and soil type. And so the way that this process, oops, the wrong direction, sorry about that. The way that this process worked is, an early farmer would harvest material from their field, and they would save some portion of that for planting in the next year. And that material, if the farmer moved to a new location, would then be used to seed a population of individuals in a new field, in a new location, with a new environment. And so these fields are really cauldrons of evolution. And that's because unlike much of modern agriculture, the fields were highly genetically diverse. So you had a bunch of genetically different individuals competing to survive in this new environment, and those that were capable of doing it would be progressed to the next generation. And so how genetically diverse were these early farmers' fields? So this is data from an experiment that my lab conducted where we sequenced the genome of 6 seed that had been collected from a single farmer's field in the Mariot region of Egypt. And these 6 seed, we sequenced each genome and then we compared it to ask, what is the rate of genetic difference that we find in various windows across the genome? So what you're looking at here is a chromosome, and each point is a 100 kb window. And each of these windows, we're asking, per base pair, how many genetic differences do we see on average between individuals? What you can see is you have this sort of landscape where things can be high or low. But the first observation to make here is that the average level of genetic diversity across the genome or across this chromosome is about 0.004, meaning that 4 base pairs in 1,000 differ between the different seeds from the single farmer's field. So to put that into perspective, when we compare that to human genetic diversity, that's over double the genetic diversity in humans at a species-wide level. So the single farmer's field contained more genetic diversity by this statistic than the entirety of the human species. Additionally, if we look at these peaks, this level of divergence that we see here in these regions that are highly diverged between our seed, that level is greater than the divergence between human beings and chimpanzees. And so really there's this enormous sort of repository of genetic variation in these early farmers' fields that could be used and leveraged in order to adapt to a new environment. So, coincident with this sort of high levels of genetic diversity and rapid dispersion across geographic range, we see that there was an explosion in phenotypic diversity that's associated with this after domestication. So this is an example of that sort of phenotypic diversity. This is the reproductive structure of barley, known as the spike. And we can see different sizes and shapes, lengths of awns, different colors. Most of this variation does not exist in the wild ancestors of barley. It arose after that. And so my lab's real interest is to try to connect this sort of phenotypic diversity to adaptation to different environments across barley's range, and to link that to the genetic differences that we see at a genome-wide level. And so, sorry, I keep getting the direction on this thing wrong. So we have sort of a few core questions that I'm going to try to address today. And so the first question that I'm going to talk about will be how important is the process of local adaptation in shaping the patterns of genetic diversity amongst early barley varieties. And the second is how much of the genome was involved in this process? Is selection affecting the entire genome? Is it affecting just specific regions? What are the sorts of traits and genes that were important in this process of adaptation? And then finally, once adaptation happens, how do populations move into a new environment? Is this a dead-end process, or can populations recover from strong genetic divergence due to adaptation? And so you might already be thinking that this sounds like something that's very difficult to study, right? So if we think about microorganisms, people who are interested in studying the process of adaptation, they typically will do an experiment where they grow cultures, and they'll do this over time in perhaps different environments. These are called this is called experimental evolution. And we, of course, can consider doing this in crop species, but in large multicellular organisms, the number of generations we can do in a reasonable amount of time is relatively small, right? So most of the people who come to my lab are interested in publishing a paper within the next couple decades. So if we want to, look at decades of evolution, it would be difficult to do by doing our own experiments. But what my lab relies on is really the brilliance of breeders who were studying barley in the early part of the last century and experiments that they set up that we can leverage with modern tools in order to understand this process. And so that's what I'm going to talk about today. And so these are two of those breeders, Harry Harlan and Mary Martini. And they were barley breeders at the beginning of the last century. And this is really the time, when we're transitioning from historical growth of historical varieties in different locale, but to a controlled breeding in genetics-informed environment where people were doing crosses, trying to generate hybrids from material collected from different places that may have two different types of beneficial traits and to combine them into a single lineage. And so Harry Harlan specifically went around the world and collected a large number of barley varieties. And what he decided he wanted to do was to start an experiment where he intercrossed a large number of varieties to generate a huge amount of material that he could do selection from in the future to identify superior varieties for the traits that he was interested in. And so he chose 28 different varieties shown here that have a global distribution and represented the sort of the phenotypic diversity that he thought was important for barley. And then he and Mary Martini initiated a crossing scheme, which involved the intercrossing of all 28 of these varieties at least once to every other one of the varieties. So this is the kind of enormous crossing scheme that apparently was possible at the time. He must have had significantly less administrative burden than I do. Anyway, I wish I could sit in the field and cross barley all day, but unfortunately, the UC doesn't allow me to do that. But this enormous crossing scheme then resulted in a large number of F2 segregating families. And in Harry Harlan's book, he actually admits that he was too lazy, or he says anyway, perhaps jokingly, that he was too lazy to maintain each of these families in large population sizes to do selections from each. And so what he did instead is he took these offspring from these large number of crosses and he pooled them all together. And he pulled them all together and began growing them out with very little human manipulation other than planting them and harvesting them in the spring in Davis, California. And so, so this is shown on the right where you take these seeds, you put them in the ground, you wait around until seed set, you harvest, you use that seed to found the next year's generation. And so the idea was that all these genetically different varieties, so tens of thousands of different genotypes are now competing in Davis, and those that would survive in that environment would be more useful for his breeding program. He would do selections from that in the future. I know there's a lot of breeders here, and they probably already know some of this history, but just to make sure everybody's on the same page. And so what makes this experiment really amazing is that through the efforts of not only Harry Harlan and Mary Martini, but also several other small grains breeders, specifically Coit Sunison and Gus Wiebe, as well as the efforts of population and quantitative geneticist Bob Allard, this experiment was maintained over the course of the last century, over into the 1980s, and was passed through over 50 generations. And because this experiment was done at Davis, and I did my graduate work at Davis, I knew this material exists, and I thought it would be a cool side project to say, maybe find some dead seed that was lying around somewhere, using modern tools to do some genomics to see what sorts of genes might be involved in this process of adaptation to Davis's climate. And so I was curious what happened to this material, and I eventually got put in touch, after asking around a lot, got put in touch with Cal Qualset, who's a retired wheat breeder at UC Davis. And he said, oh yeah, there's dead seed from that material. My department's been trying to get me to get rid of this material forever, and I was just about to throw it out. So if you want some of it, have at it. And I said, please don't throw it out. And I basically, the next day, I went up to Davis from Riverside, and he led me to the seed storage facility there and showed me these baking powder cans that they used to store the seed in. You can see the year here, 1941, that this particular can comes from. And when you open these cans, you have this beautiful demonstration of evolution where early generations are shown on the left, moving to later generations on the right. And over time, this black-seeded variety, or this black-seeded genotype that segregated at about 14% of the population, slowly goes extinct. And so what makes Cal really the hero of this story, though, is he kept this material alive. So he continued the experiment, keeping live seed. And so we can do experiments like this, where in the same field at UC Riverside, we can plant side-by-side varieties that are separated by over half a century of evolution, and ask what sorts of genetic changes occurred and what sorts of trait changes occurred. So we can begin to ask these sort of fundamental questions that are inaccessible if you don't have the ability to directly observe a process, right? So the first one was, how important has local adaptation been in shaping the patterns of genetic diversity in the population? Is most of the stuff going on random chance, or is it adaptation to the Davis environment? And so the first thing to do was to sequence the genomes of the parents of the composite cross. So this experiment was called the composite cross. I can't believe I didn't mention that. So this is the composite cross 2, and it had 28 parents. So we sequenced the genomes of these parents, and we sequenced them to sort of low, medium coverage. We identify an enormous number of genetic differences that's segregated between these founders. And then if we conduct a principal component analysis of genetic diversity, as shown over here, so what the idea here is that we're looking at the genetic relationships between these lines in a two-dimensional space. And so the dots that are closer to each other are genetically more similar to one another, while those that are farther away are more distant. What we see is in the large black dots is that the genetic space that's covered by the large black dots, which are the parents, is very well covers the global space shown by these smaller dots from a panel of global sequencing that had already been done. And so what this means is that the parents really well covered the genetic diversity that we find at a global level in barley. So the barley breeders, perhaps not surprisingly, really knew their organism. They really selected a panel that represented global diversity. Indeed, the parents, just these 28 parents, segregate for 96.4% of the common variation found in barley species wide. So we have this enormous amount of genetic diversity that founded the population. And so then we wanted to ask so we have this 13 million sites. What was the trajectory of evolution at each one of these sites over time? And so to do that, we've sequenced 3 pools of 1,000 individuals sampled from, I put 4 time points, but the pools are actually from 3 time points, F18, F28, and F58, and we have the founder allele frequencies here. And so we collect tissue, pull it together, generate a sequencing library, and then for each of these 13 million SNPs, we say, what's the allele frequency over time at these different time points? For this, we do a bit deeper coverage to get better allele frequency estimates. And so on the left, I'm showing you sort of a global summary across the whole genome of this data, where I'm on the x-axis, I have the generation of the experiment, and on the y-axis, I have estimator of genetic diversity across the founder sites. And so in the solid black line, I'm showing the observed data where we start with some level of genetic diversity, and this very linearly and very rapidly decreases over time. It actually decreases so much that within the next basically 14 generations, we would predict at this sequencing coverage, the entire population is going to be genetically homogeneous. Now, of course, there could be very rare stuff in there, but most of the genetic diversity has been removed. And so we can then the next question is, okay, so genetic diversity being lost over time is expected and can have numerous causes. One of those is just random chance. We have 2 alleles at every site, and we sample that, and that sampling process over time will result in loss of one or the other allele if you do it for long enough. This is called genetic drift. And genetic drift happens at a rate that is predicted by the size of the population. Larger the population, the less likely that sampling process is going to result in the loss of an allele. And so what we wanted to ask, is it genetic drift that causes this linear reduction in genetic diversity over time, or is there something else going on? And so what we did, the beauty of this experiment is that we can simulate at various population sizes the experiment, because we know the conditions and the way that the experiment was grown. and then ask, is this rate of loss of genetic diversity over time, is it consistent with the way that the experiment was grown? And so here we have 3 different simulations of populations. The population was actually grown more around a census size of about 20,000 individuals, but it's losing genetic diversity basically as if it were 2 orders of magnitude smaller than it is. So it's like they had done an experiment where they just grew 100 individuals every generation. So it's losing genetic diversity at this extremely fast rate. And what this strongly implies is that natural selection is playing a role in removing genetic diversity over time. And so then that left us to ask, so what sorts of natural selection processes were important in shaping diversity in this population? And so that we had 2 basic hypotheses here. So the first is that perhaps deleterious variation is involved. So our genomes, as well as the genomes of barley lines, have accumulated mutations over time. And those mutations are going to be largely either not have much effect on our biology or have slightly deleterious effects. So they're just sort of a little bit bad for us. And you have millions of mutations like that, they can accumulate to have some fitness consequences. And so that's one hypothesis, that these barley lines have accumulated deleterious variation and that it's being removed over time. So selection is removing this and leaving just those individuals that don't have that variation any longer. And so if that were the case, then we would expect that all these lines, all the parents carry about the same amount of deleterious variation. And so there shouldn't be sort of favoritism of where the alleles come from. However, if the population is evolving to survive in this specific environment, then we might expect alleles from parents that were drawn from a similar environment, that those would do better over time. And so we wanted to distinguish, is it local adaptation or deleterious variation that was important? And so what I'm showing you here is MDS analysis of genetic diversity, which is very similar to the principal component analysis I showed you before. However, I now have the population and its trajectory of evolution over time genome-wide. And what we see is that the population starts at a sort of intermediate position amongst all the parents as expected, given the design of the experiment, and then shows this extremely rapid evolution in one direction towards these orange-colored dots. And these orange-colored dots are all drawn from Mediterranean environments, which are very similar to the Mediterranean environment in which the population was being grown. And so genome-wide, we see that alleles that were drawn that come from parents from the Mediterranean were strongly favored in this experiment over time. All right, so we see this strong shift in the pool-seq data, but it doesn't really tell us much about how these beneficial alleles were perhaps assembled into individual genomes. And so the next thing we wanted to do is look at individual genotypes. So we did what's called a genotype by sequencing experiment, where we took individuals and we scored them for a subset of the markers that we had identified in the whole genome data. And so when we do that, we identify somewhere around 200,000, 250,000 markers that we can use to understand how the genomes of these individual samples looked. And we sequenced 878 different progeny drawn from across 4 different generations. And so the first thing we wanted to do is confirm our pool-seq data that genetic diversity was getting smaller over time. It would be bad if we didn't see that in the individual data. And so here we have the start of the experiment, and what I'm showing you is the genetic distance between individuals in the population. So we just compare each individual and we ask how similar are they at the SNP sites. And so we see that this value is sort of spread out, but high on average at the beginning of the experiment, and it rapidly drops over time, right? That is consistent with what we previously saw. But what was surprising about this is the degree to which how small this value is. Basically, this is at the error rate of the assay that we use, meaning that these are more or less genetically identical individuals that we're identifying in this range. So we can collapse these genetically identical lineages, and then we end up with actually, though the experiment was started with 15,000 different genotypes, we end up with a much smaller number of unique genotypes in the experiment. So of these 878 individuals that we sequenced, we identify that over 300 of them, or more than a third of them, come from just one single genetic lineage, meaning probably one individual F2 gave rise to all of these individuals. There are a number of other common lineages, though not as common as this most common one. And then, of course, we do find some rare lineages as well. So this very common lineage, we named Lin1, and then we traced the frequency of this highly adapted lineage over time. And what we see is that we can detect it a little bit by F18. So at the 18th generation of the experiment, there's a few individuals from this population, but it rapidly rises to basically be compose over 60% of the population in the later generations. So again, we started with 20,000, 15 to 20,000 different genotypes, and we end up with over half the population being derived from just a single genotype, clonal lineage. And besides, like, just in my opinion, this being super exciting for us, it was also exciting because this sort of mirrors what people have observed when you go out into wild populations and look at genetic diversity. So this is one such study where people have genotyped a large number of Arabidopsis that have been collected from various places in southern Germany. And each of these Venn diagrams, the size is how many individuals they scored, and the colors represent how many different genetic lineages they identified at these different sites. And what you can see is that most sites are dominated by one or perhaps two lineages, right? So really, an individual stand of Arabidopsis will have mostly just one genotype or just a couple. And however, why I think this is interesting is that most people assumed that this sort of pattern in selfing arises because of random chance, right? So some seed gets to a place, grows there, and then dominates that area, and that's why you get one genotype there. However, if you really think about that, Arabidopsis has lived in this place in southern Germany for tens of thousands of years, and it's moved very easily around the area. And so what our data says is that, at least in the case of the composite cross, we can start with very large numbers of genetic lineages, and we still end up at the same point. So this suggests that selection, not random chance, may be involved in this sort of pattern, or at least provides that as a possibility. All right, so in general, though, I think that the answer to this first question is in the barley composite cross, adaptation to local conditions in Davis, California was by far the predominant force that shaped genetic diversity in the population. So the next question that we wanted to ask is how much of the genome is involved and what sorts of genes might be driving adaptation? And so to do this, we compared this early part of the experiment, the beginning part and the start site, and then the F18. And the reason why we compared this early part is because at that point, we're still watching selection happening, the process of selection happening, rather than the endpoint where basically the whole genome is homogenized. So we can do what's called a selection scan. Here on the x-axis, I'm showing you the chromosomes of barley in these different colors. And on the y-axis, I'm showing you a statistical significance of a selection test. So what this means is we're looking for the characteristics of directional selection on nucleotide diversity in these regions. So when you select for a particular allele, you expect large changes in allele frequency, as well as a reduction in genetic diversity at that site. And so we're using that in a statistical test here and comparing that to simulations in order to calculate what it was likely to be targeted by natural selection. And we identify about 2.6% of the genome that shows very, very strong evidence for directional selection in the population. This is about 58 in Harvard in about 58 different regions of the genome. And so when we look at these regions of the genome, we look at the genes that they overlap, we were happy to find some of the most well-characterized barley diversity genes overlap these regions. So for example, this gene, VRS1, confers a major floral dimorphism in barley inflorescence development, so 2-row versus 6-row barley. If those people who have never heard of this dimorphism before, if you look at the size of like sort of a fancy beer, sometimes they'll actually say what sort of that is. It's usually almost always 2-row, but they'll write it there anyway. This dimorphism confers a difference in the number of rows of seed in the inflorescence. And at the beginning of the experiment, there's about a quarter of the individuals have alleles that confer this 2-row phenotype. By generation F18, the experiment is completely 6-row. So 2-row has gone completely extinct. And so the reason for that, one can hypothesize, but I think that's more for a conversation over beer. But I will mention that of the 58 regions, we identify a large number that overlap with genes that are pretty well characterized for having a role of inflorescence development in general in other systems, either rice or Arabidopsis or in other grain crops. because barley inflorescence development hasn't been studied as deeply as a system like Arabidopsis thaliana, some of them are not known function in barley, so I can't say for sure that they're involved in that, but that certainly gives us a lot of candidates to begin studying on a molecular side. But what this suggests is reproductive development may be a target for selection, right? And it's actually targeted in two different ways, because the other class of genes that are well characterized in barley, that play a role in barley diversity, that we identified, are those that are involved in the timing of heading or flowering time. And so two of the most important regulators of flowering time in barley are these two genes, PPDH1, HV-sen. PPDH1 confers the major difference between barley lines in day length sensitivity. HV-sen is a gene that, when mutated, confers earlier flowering irrespective of environment. And so so when we think about flowering in this composite cross experiment, it sort of has a natural connection to adaptation. That is, in Mediterranean environments, and the barley composite cross specifically, were grown by planting in the fall, allowing overwintering, and then harvesting in the spring. And an important potential selective factor is that in Mediterranean environments, you have this very low amount of precipitation in the summer. And so what that means is that if you don't flower by the time precipitation ends, then you're going to go extinct. And so this is so the model here would be truncating selection, where if you flower too late, you're out, right? And so to ask this question, so alleles at both of these loci are Mediterranean alleles that confer earlier flowering, and so that's consistent, right? And so we wanted to measure flowering time and see if this was actually the case. And so here I'm showing you data of F18 and the early generations where we're comparing flowering time, and we do see a shift in about 7 days of earlier flowering. And in particular, we see the removal of these very late flowering genotypes. So we don't have really late stuff that would be past this precipitation gradient. All right, so great. Directional selection on this process, sort of neatly tied up story, right? Well, the beauty of this population is it lets us to really observe these traits over longer periods of time. So I just told you this story where we start with a wide distribution of flowering times. We're off the fitness optimum where we want to flower right here. And then over time, selection moves this distribution of flowering time to an earlier time point through PPDH1 and HV-sen, perhaps some other genes as well. However, here's where the long-term experiment really becomes favorable. So what we see is this drop in the initial generations, but then the population just turns completely around and evolves a later flowering time, actually a slightly later flowering time than the initial flowering time. And so so what we thought was directional selection actually really looks like an asymmetric form of a different kind of selection, and that is stabilizing selection. And so the most famous case of one of the most famous cases of stabilizing selection is that of birth weight in humans. And the idea here is if infants are born with very, very low birth weights, they tend to suffer mortality at a higher rate. Or if they have very, very high birth weights, they also suffer mortality at a higher rate. And so that drives the distribution of this trait value of birth weight towards a sort of intermediate value. And it's actually known that because of modern medicine, or correlated with the developments of modern medicine, this truncating selection at this side has been relieved a little bit. So it may be that because higher birth weights don't cause maternal and infant death at the same rate as they used to, it may be that we have relieved some of the selection pressure. But nevertheless, this generates a narrowed distribution over time. And that's exactly what we're seeing here, right? So we start with truncating selection at the end of the season, which removes these outliers here, and then these outliers are removed over long enough time. And so this is actually predicted by population geneticists in the past who modeled what's called an asymmetric selection gradient, so that this side, although this side is really bad, you die, you don't make seed. If you flower too late, you're not going to get harvested. However, this side, it's not good to be here, but perhaps the selection pressure isn't quite so strong. And so it takes a longer time to actually observe that difference. And so it's really that asymmetric selection coefficient which gives us this time. But nevertheless, if we hadn't studied this long population, we would have just shut the book and said, we have directional selection on flowering time, and that's the process we're studying. But instead, we have this much more, in my opinion, interesting process that hasn't been studied much from the standpoint of molecular genetics. And the other beauty of having segregating variation at this stage is we can do GWAS because we've genotyped all these individuals, and then we can ask what's the genetic determinant of this second shift in phenotype. So when we do GWAS in the progeny of the composite cross, what we see is a single peak on chromosome 4, which overlaps this another well-characterized flowering time regulator called VRNH2. And when we look at the VRNH2 locus, we identify 5 different haplotypes that initially segregate in the population. But basically these, and 2 of these are earlier flowering, and 3 of them are later flowering haplotypes. And the population in late stages have been driven to basically only be composed of these later flowering individuals. So if we look at the VRNH2 locus in more detail, here VRNH2 is conferred by the deletion, the phenotype of VRNH2 mutations conferred by the deletion of 2 excuse me, adjacent genes. And this deletion is present in the early flowering types. And so you can see here, this is a coverage plot where we're looking at aluminum sequencing coverage over this locus. And we have basically no coverage in these 2 early flowering lines. And we have coverage in these 2 haplotypes that are late flowering, we find coverage. And in the population, over time, we see that this late flowering functional alleles have been maintained. And so we can directly connect this second shift in flowering time to the retention of VRNH2 function over time. So again, we start with this wide distribution of flowering time. It's off the fitness optimum. Then these earlier flowering alleles at PPDH1 and HV-sen are driving this distribution towards earlier flowering, but that earlier flowering phenotype is then modified again by a second locus, VRNH2, narrowing this overall fit distribution of trait variation. All right, so the answer to the second question was how much of the genome is involved. We think at least the strongest selected loci, it's about 3% of the genome. And what sorts of genes and traits are involved, it seems that reproductive biology has been central to adaptation in this particular environment. And so in this last part of the talk, what I'm gonna talk about is the process as whether this is a dead-end process. So are we looking at a process that sort of fixes these genotypes in a particular state? So remember, we started with a lot of diversity. We end up with almost none. And so that's a little concerning, right? If we think about that, if we think about these populations here, for example, in the wild species, where they've basically been fixed for one genotype at all these locations, what happens when the environment changes, right? So if we have a subtle or not so subtle shift in these environments, does that mean all these selfing populations are just going to be wiped out? Do they have the ability to survive if that happens? And so I'm not going to answer this question in totality. I think it's a really big question. However, I do think that an experiment tells us a little bit that they did with the composite cross tells us a little bit about this. All right, so this is work that's done by a graduate student in my lab, Jill Marzolino. And what these folks did is that about generation F24, they took a batch of seed and they shifted it to a totally new location. They began growing the population in parallel at this new location. And this new location is Bozeman, Montana. And I suspect most of you are aware that Davis, California and Bozeman, Montana have slightly different weather. In addition, they grew it in totally different conditions. So in Davis, this was fall planted population. In Bozeman, the way you grow barley is by, at least as I understand it, is largely by spring planting. And so this population was planted in the spring. And so this is a massive environmental change, right? And so we already had removal of a huge amount of genetic diversity in this population. And now we're challenging it with this major shift in environment. Can it respond to this environment and how? And so we did the same sort of experiment. What does genetic diversity look like over time? And so here I'm showing you the Davis population where you have the strong linear decrease. And the first thing that happens in Bozeman, which is shown by this dotted line, is genetic diversity basically crashes in the first few 3 generations. And so that crash makes some sense, right? If you take a bunch of lines that are adapted to one environment and you plant them in a totally different environment, even if you don't assume there's any adapted lines in that initial population to the new environment, you would expect things to just be culled out by random chance, like only a few things are going to survive. And so a loss of genetic diversity here makes some sense. But the second thing that happens is that we have this massive skyrocketing of genetic diversity over the course of the next up by generation F50. And I did not believe this result. So I told Jill, showed me this result. I was like, you mixed up the samples. You got to go back and do this. You got to do it again. She did it again. I said, I don't believe it. I think we must have ordered the wrong things or mixed up our initial stuff, which she wouldn't have done. So I didn't blame her on that. That was other people's responsibility. So we ordered it again. We did the experiment. We got the same result. So it's really that we have this very high level of genetic diversity in this later generation. So why would that be? And so this is only a partially finished project, and so I won't tell you the entire story, but I do think that we have a pretty good idea. I'll show you one result which tells us a little bit about it. And so here I'm showing this MDS analysis where we're looking at directional change in Davis. So we have Davis population starts in the middle, and then over time it evolves towards these orange Mediterranean lines that are. So what happens to Bozeman? So, oops, sorry. So in Bozeman, what we see is that initially in the first 3 generations, the population kind of evolves along the same trajectory. So not enough change has been, so basically you have a reduction in genetic diversity, but what's around is still mostly stuff that's adapted to Davis. It's similar to the Davis environment. But then between F27 and F50, the population completely reverses course. So it now turns around and starts evolving towards these lines in the other corner. And these lines are the northern Eurasian lines, which are adapted to cold environments and also more often grown in the springtime and have flowering times consistent with that. And so what seems to be happening, and the reason why you get higher genetic diversity, which we've confirmed more recently with individual sequencing, is that there were very, very, very rare sort of Eurasian type lines that existed in the population the whole time. They were never completely removed. And that these rare lines are now sweeping back through the population. And what we see, we're not at the end point, which is like genetic homogeneity. We're in the intermediate point where there's both Davis types as well as Bozeman types around. And so this looks like really, really high genetic diversity from a statistical standpoint because everything's basically at an intermediate frequency at that point. So the skyrocketing diversity is an intermediate point, presumably towards, again, fixation towards things that are more like the Davis, the Bozeman, that are better adapted to the Bozeman environment. And so I find this sort of encouraging because it means that stuff that exists in populations and selfing populations at very, very low frequencies can relatively rapidly, possibly partly because of how selfers work, resurrect basically after some major changes. Now, these populations in the real world may not be so carefully maintained at high population sizes. However, when we think about selfers in the wild, they do have very large seed banks. And so if these sorts of genotypes remain in seed banks, then even after change, these populations may be able to survive shifts. Or at least it's not impossible, which is a good thing, I think. All right, so that's really the last bit of research I'm going to talk about. Again, I would just want to acknowledge the people who do the work. This is actually a recent lab event for the graduation of Chris Fiskus, who's a graduate student whose work I didn't get to talk about today. But this is the lab, both my lab and Danelle Seymour's lab, my partner and also a professor at UC Riverside. They work on citrus. If you're interested in that, I can point you towards her. This is really just the beginning. We have a huge amount of material from these experiments. Probably my favorite experiment that they did is they planted the entire spring collection of 6,000 different barley varieties, every other row with male sterols, and generated a population that could have as many as 6,000 different parents. But they did these long-term experiments at multiple sites with different genetic compositions. So there will be a lot coming in the future. And then we've also been doing a lot of our own work looking at phenotypic differences across larger numbers of scales, but not on roots yet, but on aerial traits at least, as well as inflorescence morphology and work that I didn't have time to talk about today. So with that, I'd like to thank you all for your attention. And if there's still time, I'd be happy to take questions.
This basic question.
Yes.
So is there no chance whatsoever of outcropping?
So there's not no chance, although they were pretty careful about that. We can model what outcrossing would look like. So especially for the Bozeman data, I think that that's the most what I would be slightly concerned about. Basically, because we know the parental alleles, we do detect if there is something like totally unusual coming into the population, but we can't know if something very similar to one of the parents is introgressing. But we can run our models with introgression and see if this amount of change is likely under like insane introgression rates. So it's known that barley outcrosses about 2% of the time. They did a large number of experiments before starting this to actually establish the outcrossing rate in barley. Yeah, they did that is true, although I don't know if there's a lot of evidence that increased outcrossing results from very many environments. There's certainly the opposite has been shown many times. But nevertheless, it is possible that some gene flow comes from elsewhere. I don't really think that the only thing that changes is really the last part of the experiment. So unless you assume extremely high levels of outcrossing with whatever's around, which they also tried to prevent that from happening physically. And they calculated the sort of rate, like how far pollen is expected to go. But even if we assume like sort of reasonable levels of outcrossing, you wouldn't get this level of change in simulations. You would have to assume like really, really high levels of outcrossing for that to happen. This is the same, and that problem is something we looked at quite a bit because it's the same with seed contamination, right? So if seed contamination happened some point, we don't have a lot of evidence for that, but if it did happen, you would have to assume that it would be like, the entire population was just mixed up. Given the geneticists involved, I think that's very unlikely. Also, just to remind everybody, they were breeders and they were maintaining varieties, like many, many hundreds of varieties purely for many long periods of time. So I think that they had a pretty good idea of when such contamination happened, but it's always a concern. So we do simulations to try to ask that question. So yeah, it's a good question. Yes.
So really,
Thanks.
So with GWAS, a lot of the time, or I guess most of the time, you're looking at allele frequency. So you're kind of looking sort of like a haploid perspective. But if you look at genotypic perspective, how much does heterosis play into account?
Yeah. So I love the idea that heterosis might be playing a role. That we just discussed the fact that not too much outcrossing happens in barley. However, the first paper that was published from a population genetic standpoint in this population showed that some loci in the genome are maintained as or that heterozygotes are more often found than you would expect given the outcrossing rates. Not all sites, so it's not a contamination issue, but just some of them. And what this was used to argue for was for high heterozygote advantage, which is, probably some of you may have heard of heterozygote advantage. So for example, when we think about sickle cell anemia, the genetic determinants of sickle cell anemia result from a selection pressure for malaria resistance. So that same, if you're homozygous for one allele, you get sickle cell anemia. That's not super good. If you're homozygous for the other allele, you are susceptible to malaria. Well, if you're heterozygous, you're less susceptible to malaria and you don't have sickle cell anemia. So this is called heterozygote advantage. It's a very famous case of how genetic diversity can be maintained in populations. However, this is one of these things that, every field has something they argue about forever. And this is one of those topics is like how often can single locus heterozygous advantage actually confer increased fitness? And then for breeders, how often does single locus heterozygosity confer hybrid vigor, right? So these are the same argument that are had in both fields. So in this case, that what they're arguing is that it does happen sometimes. And so they did see evidence of that, that being, that being a hybrid was actually beneficial. But of course, or it happened more often than you would expect by random chance, at least. And so that was a paper, I think that was a Nature paper in like 1960. So yeah, so it's possible. Yeah.
What about static interaction?
Yeah, I love this idea. Yeah, I think so. That is one of the cool things about this experiment is that we can actually probably try to get at that. But I don't have any answers for you at this time. But we can, because we can take the material that we know is adapted, and then we can do intercrosses, and then ask things about like what's the distribution of genetic sort of fitness effects across loci. I think we can actually ask that question, but I don't have any answer for you at this point. I think it's a great question. I would love to study that. Yeah.
Individual weather events like a wildfire or the general trend in global warming.
Man, you should write my grants. Yeah, so we don't. So global warming is something that people often ask. The populations that we're working with, the way that the population was grown makes it a little bit difficult to study that specific problem. But what a postdoc in my lab, an experiment that she sort of did at the very end of her postdoc, is that these populations were actually grown in parallel over many years, every year between 1970 and 1980. And so we produced this huge, we sequenced 100 individuals from every generation from multiple parallel populations. And the idea was that we would use the local weather station data from Davis to look at, so California has a cyclic climate. So that's why you hear like, probably, at least if you're not Californian, you hear that like alternating things. One, like it's a horrible drought, everybody's on fire, and then the other years it's like everybody is flooded. And that's because we have this cyclic pattern of weather. And there's been shifts in that pattern recently, which has made the droughts worse than they used to be. However, it's always happened that you would have dry years and wet years. And over this period of time, we have that pattern. And so we can look at with yearly resolution, is there a correlation of the success of particular alleles over time and that yearly weather pattern? And so that's exactly the kind of thing that we have the data set, but I don't have anybody to work on it because she got a job and went to do something else. Yeah. Uh-oh. Okay. Mm-hmm.
Seems like your mutation set is finite in a way.
Yeah. Mm-hmm. Yeah, so that's a really cool question. So the question was, since it was somebody in the back, like, what is the role of epigenetics in adaptation, especially given that you only have a set amount of genetic variation at the beginning? So the short answer is we don't know. I think that is something that we would be very interested in studying. I worked on epigenetics as a postdoc a bit. I tend to be on the side that when we're talking about natural populations, that most major changes in epigenetics are driven by genetics. So, either a change in transposon distribution or a change in mutations of important regulators of epigenetic processes. But I could be totally wrong about that. So it's definitely something we want to study. We were going to do, sort of the transcriptome first on the functional genetic side. I have money to do that. But epigenetics would be a really interesting thing to do as well, I think. I should say also, at least in Arabidopsis, there's between Arabidopsis and many selfers have the same sort of gradient of northern Eurasian and Mediterranean types with different suites of phenotypes and different growing happens. So there's a lot in common between barley and Arabidopsis from this standpoint. And there's a big difference in Arabidopsis in sort of overall DNA methylation rates in northern lines compared to southern lines. So it suggests that at least in that system, that there's something different about those two environments that causes that. Yeah, multi-layered question. So yeah. Hooded is very Yeah, hooded is like very deleterious. So hooded and also free threshing in barley, all these, they don't survive. So it's like from a competitive standpoint, those are much less competitive. I think people have known for a long time that in barley, for some reason, well, there's some hypotheses, so I don't want to say anything that people have. I don't want to say anything that might insult anyone, but so I don't mean to dismiss that work. But basically, barley is unlike wheat. If you have these free threshing types, they tend to have much lower yields, and they also have a much lower fitness in this population. So those don't survive at all. We haven't looked in. I don't think we've seen any hooded or so. One of the parents was hooded. One of the parents was free threshing. I didn't even say that. Sorry. Several of the parents, so a quarter of the parents were two-rowed. So these traits were all segregating in the initial population, but most of that, so the black-seeded type goes extinct, two-row goes extinct, hooded goes extinct, as does free threshing. And so some other change traits that are known to change, so the seeds get bigger over time, the seeds get substantially bigger. Yield from the beginning of the experiment to the end of the experiment almost tripled. But I should say that it didn't keep up with selected lines by breeders, which is why this evolutionary experiment thing, which went on for many years at Davis, didn't really work out from the standpoint of producing new varieties. So from an evolutionary geneticist standpoint, I think that's one of the most interesting things about the whole experiment, because the reason why, though I know there are people who say they know why this is, but I think it hasn't really been dissected as much as it could be. So the reason why these, sort of things that are selected to yield well together as a homogeneous population, why these are less competitive than things that are grown in this particular environment. I think that there's a lot of fruitful area for discovery. Yeah.
Farming practices, harvesting technologies change with that 58 that influence selection of what.
So of course that's a possibility. Most of the experiment was conducted by Wiebe, and Gus Wiebe would do hand harvesting for everything. But in more recent years, as we continue the experiment, it's likely that the more recent years have been done with combine. There's no like major shift that's associated with that, but it is definitely possible that would result. This is also important for discussing what you were talking about, the contamination issue, because he was hand harvesting. This issue is significantly less of a problem than if you combine harvest. Plant height massively increases. Yeah. So these things lodged are a pain in the ass in the field. So yeah.
You were focusing kind of on flowering. See a relationship between the size of flowering, so the biomass data has collected?
Yeah. So it hasn't been, from these experiments, we didn't collect it, but we have data to study that. And we're in the process. So I don't have any result for you, but we don't have field data, which would be a cool thing to do, but mainly because of the capacity for growing a large enough, like we have to actually maintain the material in addition to doing our experiments. So if we do a biomass study early on, a destructive biomass study, then like that's more than the capacity my little lab has right now. But we do have greenhouse experiments where we've monitored the entire course of development in not all the lines, but hundreds of lines, using sort of phenomics type technologies. The greenhouses obviously have their issues. Though I will say that all this flowering time data that I showed you today was greenhouse data, but we've replicated it now in several years of field studies, and we get the same exact results. So it's, at least from the standpoint of flowering time, consistent. With respect to biomass, that's a different question. So. Yeah. Yeah. So I didn't give a reason why there might be selection for later flowering, right? And I think that there's two reasonable hypotheses. One is the one you're talking about, that you need to accumulate a certain amount of biomass before you flower to set enough seed. The other hypothesis is, although weak selection, that perhaps if you flower too early, you might occasionally get exposed, some years get exposed to some frost, which does occasionally happen in California in the very early parts of the winter. So I think it's a perfectly reasonable hypothesis. That's usually what I tell people when they ask me, that's you need to accumulate a lot of biomass. And it's true that if you look in the greenhouse where there's no end of season selection, like the number of seed that are produced is very well correlated with flowering time. It's like almost perfectly correlated. In the field, it's a little more complicated, but that's the case.
5 o'clock, but we're gonna have outside questions coming after, so we're gonna end the seminar. If you'd like outside
Yep.
Thank you.
No, thank you. Thanks for the great questions too.